Okay, in that case, good afternoon, everyone. Hope you're all doing well. My name is Ido Diamant. I'm a bioinformatics software engineer in the Ion Lab, and today we'll be discussing normalization and some of the methods that we're going to use to carry this process out. Just a quick outline of what we'll talk about. First, we'll go over sometimes why we need to normalize our data, some of the biases and effects in our data that we need to correct for as we do more processing. We'll go through a few different methods of normalization and where they might be useful. And then we'll look at some actual examples using some real microarray and RNA-seq data. So first of all, before we start normalizing, we need to understand why we actually sometimes need to normalize data. When we're looking at omics data, specific, for example, transcriptomics or proteomics data sets, we can be looking at data that has large differences in scale and distribution. So if we look at this data just as is without correcting for those differences, we can actually get skewed we can be working with skewed data and draw incorrect conclusions. The point of normalization is to preserve those biological features that we actually want to observe and draw conclusions from, but remove technical bias in the process. Downstream analysis steps can be affected by this unnormalized data, especially if they take into account distances between values, like we see in differential expression analysis and clustering, but as well, enrichment analysis, network analysis, and machine learning applications can all also be affected if we feed unnormalized data in these processing steps. If just looking at some real examples, this is a geo study where we can see 4 samples with significantly lower RNA-seq counts in their libraries versus 4 samples with significantly higher RNA-seq counts. If we're looking at just the distribution of these counts and we try to draw conclusions from this data as it is, we can get errors in our downstream analysis and be looking at the wrong effects. So we want to correct for this inconsistency between samples as we move forward. Typically, the first step in this process will be some sort of log transformation, where we'll apply a logarithm to every value in our matrix. This will reduce the variance between our very small values and our very large values. By instead of looking at the differences in the values themselves, we'll be looking at differences in the orders of magnitude between those values. This is also useful to correct some skewed distributions sometimes, where here on our left, we can see this very skewed distribution with very small counts, but we have a few counts that extend all the way out to, say, 10,000. And then after we perform this log normalization, we get this much more normal distribution. This one on the left is actually called the log normal distribution for that reason, where once we normalize it with log, we get a normal distribution as a result. When we're looking at this in omics context, especially with RNA-seq, we'll get a lot of 0 values, which is an issue because if you recall, trying to take the log of 0 will give us an error or it is undefined. So what we'll typically do to correct for this is add a coefficient, usually 1, to all of our values. And then we'll take the log of that, which negates our errors that we would get from counting 0 values. This does have a larger effect on small values than large values. Taking the log of 1 versus the log of 2 will be a larger change than log of 5,000 versus log of 5,001. So we just need to make sure that we're taking that into account as well later in the process. The next method that we'll discuss is z-score normalization. This is a statistical method that you've probably seen before in some context, where for a certain series, we'll be using the statistical measures of that series to transform it into a different distribution, or to correct the scale of that series. The way we do this is by taking the each value and subtracting the mean of that series from it, and then dividing by the standard deviation of the series. And what that will give us is our z-score. For any given series that we do this for, our z-scores will have a mean of 0 and a standard deviation of 1. So essentially, we can think of our z-scores as a number of standard deviations away from what we expect to see for this series, a number of standard deviations away from the mean for this distribution. Often in this omics context, what we'll look at is doing z-scoring on specific genes rather than our population as a whole. So we'll be looking at rows in our matrix as series rather than our overall matrix as our series. Here, when we look at our data table before normalization, we can see our first row of genes has much higher values in each tissue than genes 2 and 3. So if we're just looking at the highest value, this might cause us to miss some more significant differences than we expect to see. Here, once we perform z-score normalization, we calculate the mean and standard deviation for each row and carry out that formula that we just discussed in the last slide. We'll get these values, which for each row, we'll get a mean of 0 and a standard deviation of 1. So if we see any values that are significantly higher or significantly lower than we expect to see, we can see that more clearly with these z-scores. For example, if we're just looking at the highest values for normalization, we might have missed that for gene 2 in tissue 2 and for gene 3 in tissue 4, these values are more highly expressed than we expect to see for those genes, which could indicate some sort of actual biological effect that we want to look into further. Next up, we'll take a look at how this can be applied on real data. We'll take a look at this microarray study comparing sex-dependent differences in glucocorticoid and mineralocorticoid receptor signaling in mouse hippocampi. This study had 31 samples with up to 4 replicates of each condition, which gives us this data matrix here in the bottom left corner, where for each sample, we'll be looking at intensity values for each of these 41,174 probe sets, which map to our genes or transcripts. Again, after we perform our log normalization to stabilize the variance between our very small values and our very large values, we get this distribution on the left, which is still kind of somewhat hard to see where our actual effects are that we want to look into further. So after we perform our z-score normalization for each gene, again in the process we just discussed, we get this more normal distribution where we can identify specific values that are more highly or more lowly expressed than we expect to see. The next method that we'll talk about is quantile normalization. This involves making the statistical properties of 2 or more distributions identical. So in this case, for each sample, we'll carry out quantile normalization. This is done by ranking values in each sample and then creating a reference distribution where for each rank, the value is equal to the mean of all the values for that rank across all of our samples. We'll walk through an example on the next slide that will help visualize that a bit better. Then we'll take that reference distribution and substitute the values for each rank back into our original matrix. What this does is for each rank, for the smallest value, for the second smallest value, all the way up to our mean and our maximum values, we'll have the same values across each distribution. This can be useful when we're looking at values that have these different ranges and different distributions. For example, with microarray probe intensities and for RNA-seq counts, especially when we want to carry out some statistical tests, such as in differential expression analysis, where we're assuming that our distributions are already equal. So to walk through an example of this, here on our left, we have our original matrix. I've color-coded, sorry, second. I've color-coded each row so we can sort of keep track of where these values are as we move them around and do some substitution. The first step in going from our original matrix to our quantile normalized matrix is to reorder our values in each column from smallest to largest. This will then let us get the ranks in our ordered matrix, where for each row, we'll get our smallest value, our second smallest value, and those rows will become our ranks. Then we'll distribute those back into the same shape as our original matrix and get ready to create our reference distribution and substitute those mean values back into this ordered ranked matrix. Then when we go back to our ranked matrix, we can calculate the means for each row, and this will give us our values for each rank. So in this case, when we take the mean of 2, 3, and 4, we get 3, and then we'll do that for each row, and we'll get our values for each rank. Then we'll substitute those values back into our rank matrix. Um, and then what you can see is for each column, we will get this same range of values with the same distribution. So for column one, we will go 3, 8, 6, 5, 5. But for the second column, even though those values are in a different order, we're still looking at those same values. We'll take a look at how this can affect our real data once again, and we'll see how this shifts our distributions um, as well. So now we'll take a look at an RNA-seq study. This is our example we saw earlier of our very uneven library sizes across our different samples. This RNA-seq data set is comparing 4 samples from our mantle cell lymphoma tumor cells from 4 patients, and then peripheral blood B cells from 3 healthy volunteers, and from a JECA1 MCL cell line. So we've got our 8 samples with RNA-seq counts, and now we want to normalize this data. So once again, we'll start with our log transformation. In this case, since we're working with these 0 values sometimes, we've applied our log1p transformation first off. So here on the left, we can see those log transformed values. We have some gaps and some discrete values on our left side of our distribution, and kind of a smooth distribution all the way out to our maximum values, which makes it a little bit hard to see what we're actually looking at. On our right, after we performed quantile normalization, so we've ranked each sample and computed our average rank values and substituted those back in. We can see this distribution where on our left, those sort of discrete values have been filled in, and on our right, our maximum values have also been sort of averaged across our different samples. But we also see this more clear bimodal distribution emerge, which gives us a clear idea of what we're looking for. If we look at the density of values across these samples as well, again, when we look at our log1p transformation on the left side, we see these distributions still aren't quite equal across these different samples, which, again, if we want to perform any statistical analyses, can be an issue. But after we've performed this quantile normalization, we see our distributions are matched or almost matched for all of our samples. There can be some small adjustments when ranks overlap with their values, which can lead to some of those changes that we see in the left peak. But overall, each sample follows this nice, neat bimodal distribution, and can lend itself to further testing and analysis further downstream. Next up, we'll talk about median polish. This is a method that we use to establish an additive model for a two-way distribution. So if we're looking at our matrix where we have genes as rows and samples as columns, the goal of this is to make a equation where essentially for each value, we can think of the value as a sum of the overall effect of the matrix, the overall effect of each of the row that value is in, the overall effect of the column that value is in, and then some residual value. What we're doing here is essentially we want to remove any values that are applied to all values in a row or all values in a column, which can suggest some baseline for gene expression or bias in our samples. And we want to look at just the individual differences between these gene and sample relations. What we'll do this by finding the medians for each row and each column and pulling them out to our row and column effects and our overall effect. And we'll iterate through this process a few times until these row and column effects begin to change less and less as we do each iteration. Typically for a small matrix, we'll do this 2 or 3 times. In this case, we'll work through it once. Whereas for some larger matrices, you might need to do this more, let's say 5 or 7 iterations. But we want to be careful of doing this too many times and distorting the data more than we need to when we're looking for these residual values. Again, to look for an example of sort of what we're doing for each step, we have our original matrix on the left here. And then for each row, we'll find the median value. So for our row of 10, 22, 15, 27, we'll find our median of 18.5, and we'll subtract that from all the values in the row and add it to our row effects on the left. We'll do that for each row. And then once we've done that step once, we we'll take a look at the median of those row effects and subtract that from each of the row effects and add it to our overall effect. So in this case, our median of our row effects is 32.75. So we'll subtract that from each row effect and add it to our overall effect. Now, starting from that matrix that we just did, our next step is to do the same thing along our columns. So we'll find and subtract those medians and add them to our column effects. And then we'll look at the median of our column effects, subtract that from each column effect, and add it to our overall effect. So in this case, that effect is 1.75. So we'll subtract that from each of our column effects, and that will be incorporated into our overall effect. So now what we're left with is our residual matrix, where for each value, if we add together the overall effect, the row effect, the column effect, and the residual for that combination of row and column, we'll get our original value. So again, we've pulled out our values that are consistently a factor in each of our columns and rows and each of our values overall, and we're left with this residual matrix. Next up, we'll discuss some read count normalization for RNA-seq data. As we've seen already, library size can vary between experiments and even within experiments. So we want to make sure that we're accounting for this library size as we look at these RNA-seq counts. If we've performed more quantification in a certain library, we'll get more counts, which isn't necessarily useful when we're looking at comparing between samples. We also need to take into account gene and transcript length, and that somewhat intuitively, when we're looking at a larger gene, more fragments of that gene will get sequenced than if we're looking at fragments of a smaller gene. So we want to make sure that we're taking the gene and transcript length into account as well with some of these processes. So a few of the ways that this can be carried out are with CPM, RPKM, or FPKM, and TPM. We'll start with the simplest, which is CPM, where for each count in our library, we'll normalize this by some scaling factor that's based on our total number of counts for that sample. So in this case, for each gene X, for the number of counts for that gene, we'll divide this by the total number of reads and multiply by 1,000,000. In this case, 1,000,000 is a pretty arbitrary scaling factor. It's just used with these large numbers because it reduces the number of decimal points we have to use and makes our data more readable. We'll do an example in the next slide where we replace that value, but essentially 1,000,000 is the scaling factor that is used for most of these calculations, which is why we're looking at counts per million or reads per kilobase of transcript per million mapped reads. Speaking of which, RPKM is the next method we'll discuss, which is reads per kilobase of transcript per million mapped reads. With CPM, we didn't account for our differences in transcript or gene length at all. So for RPKM, we'll actually do the same process that we do with CPM, where we divide our individual gene counts by the total number of reads for our library and multiply by a scaling factor. But we'll also be dividing by the length in kilobases of each of that specific gene. So if we're looking at a gene with a 4 kilobase length, we'll divide that CPM value by 4 to account for that larger gene size. FPKM follows the exact same process. The difference is that RPKM was developed for single-end sequencing versus FPKM was developed for paired-end sequencing, where you can get two reads that map to the same fragment. FPKM essentially just keeps track of that, so those fragments aren't counted twice. But it carries out the same process where we'll take our overall counts, divide by our transcript length and number of counts for our library, and multiply by our scaling factor. Finally, TPM, which is transcripts per kilobase million. This process involves doing the same steps that we did for RPKM and FPKM, but we'll do them in a slightly different order. Instead of normalizing for library size and transcript length at the same time, what we'll do is first we'll account for our transcript length. So we'll do that transcript length kilobase division first. And then you'll use those scaled numbers to calculate our new library size and divide by those. We'll walk through a quick example of each of these, just so you can see how these values change and how each different method gives us a different output. So here we have our original counts on the left. We can see if we look that we're looking at a sample 1 or replicate 1 has 35 reads, whereas replicate 2 has 106. So we want to normalize for that library size differences. And each of our genes also has a different length in kilobases, which we want to account for if we can. First off, CPM, which again, we're not accounting for transcript length here. We'll scale this by our scaling factor, where we take our total number of reads for our library and divide that by 1,000,000, or in this case, 10, just because our values are smaller. So for the sake of readability, we'll use 10 instead of 1,000,000. But those are essentially interchangeable. When you're looking at larger values, we'll use this larger scaling factor. So when we look at our values for replicate 1 with CPM, we'll divide 10 by 3.5, which is our replicate 1 scaling factor, to get our CPM value of 2.86 for gene A in replicate 1. We'll do that for each of our values, and we'll get these new scaled counts. Moving on to RPKM or FPKM, we'll actually do the same first step as that CPM process. So we can just use those values as our starting point, and then we'll divide by our length in kilobase for each gene to account for the differences in gene length. So starting from 2.86 for gene A in replicate 1, we'll divide by our length of 2 kilobases, and it'll give us a new RPKM value of 1.43. And in this case, we can see that even though we had significantly more reads for gene 2 in replicate 1, we're actually looking at the same proportion of counts once we've normalized for library counts and gene length in each of these samples. And we'll repeat this process again with replicate 2 with our scaling factor of 10.6 and then dividing by those same gene lengths. Then we will talk about TPM, just again, an example of this to see how we're carrying this out, where our first step is adjusting for this difference in gene length. So first we'll divide our original counts by our kilobase length for each gene. And then we'll sum those up to make our RPK, reads per kilobase scaling factor. Again, we'll divide that by 10 in this case for our smaller counts to get our our scaling factor for each replicate. And then we'll divide those original counts by the scaling factor. So we can see now with TPM, though we did the same steps as RPKM, this doing them in this different order has given us different values, which can be useful for our comparisons across these samples or within our replicates as well to see different values of different genes. At this point, TPM is the most standardly used. So this is often what you'll see these normalized counts in. And then the final normalization method that we'll discuss and walk through is the DSeq2 library normalization method. A lot of differential expression analysis packages that we'll use will have some sort of normalization method built into them. So when they take raw counts in, they're doing some normalization and then performing the differential expression analysis. In the case of the DSeq2 library, we're using a median of ratios method. What this means is that we will find a column-specific scaling factor for each of our samples that takes into account both our library size differences, but we'll also be looking at library composition differences. What I mean by that is essentially when we're looking at two samples that we want to compare, say one from spleen tissue and one from liver, we'll expect to see a lot of liver-specific genes expressed in the liver, but not so much in the spleen sample. So if we're just accounting for library normalization, those reads will kind of get soaked up by other genes. So what we want to do when we're making these scaling factors is account for, again, our genes with 0 counts, as we see this library composition, and downplay those genes that soak up the additional reads. The way that we do this is starting with our original count matrix. We'll take the log of each count. In some cases, this will give us an error where we have a 0 count, which we'll take into account in the next steps. Then for each row, for each gene, we'll take the average of those logs, and we'll drop any of the rows where we had one of those 0 counts, which means that they won't be considered as we're making these column-specific scaling factors. The next step is to take that matrix. Again, we're omitting these rows where we had 0 counts, and we're going to subtract the average logs that we calculated in the last step. So for gene 2, that's 1.7, and for gene 3, it's 4.3. And we're left with these new values. Then we to actually make these column-specific scaling factors, we'll take the median value for each column, and then we'll undo the log transformation that we did earlier. So in this case, for our median for sample 1, we get minus 0.9, which when we raise e by minus 0.9, we get a scaling factor of 0.4. We'll do that for each of the samples. And then we'll go back to our original count matrix and we'll divide our original counts by the scaling factors that we've now calculated for each gene, or for each sample, not for each gene. What you can see is that we preserve our zero count, but we haven't soaked up those reads too much into other genes. But we've also scaled for library size, so now we're looking at the more comparable values between our different samples, which will be more reflective of actual changes as we perform more analysis steps. So far we've looked at the effects of our gene and sample relations as a measure of our normalization effects. But another method that is useful to look at are correlation coefficients between pairs of genes. So essentially, from our gene sample matrix, we can take pairs of genes from our matrix and calculate the Pearson correlation coefficient between them, which gives us a measure of the strength and direction of the correlation between two genes. The reason why this is useful is that typically when we're looking at unnormalized data, we'll expect to see more of a skew of those correlations, versus after we've normalized our data effectively, we can see a more clear correlation distribution that we expect to see, usually centered on zero, reflecting that most genes are not significantly co-expressed in these samples, although they appear to be initially. So here we'll take a look at exactly that. Here in the blue, we have our raw counts, our raw Pearson correlations, which we've calculated by taking 1,000 random samples from ARCGIS 4, and taking the top 5,000 genes from those samples and calculating our correlation coefficients. When we're looking at these unnormalized correlations, we can see we have kind of a skewed distribution with an average value of 0.6, signifying that a lot of the genes that we're looking at are significantly co-expressed. Again, we're looking at a range of -1 to 1 for these values, so this gives us sort of a measure of what we expect to see. Because of our systematic bias in these samples, we can draw spurious conclusions if we're looking at these gene correlations, which can be introduced by different library sizes or a number of other factors, as we've discussed already. But again, we want to make sure that we get this distribution that's centered on zero after we've performed effective normalization. So in this case, we've done our log transformation and our quantile normalization. And now we can see our correlation distribution centered on zero with a more reasonable range of values, as specific genes actually have some significant co-expression. The last new topic we'll cover is single-cell RNA-seq normalization. As single-cell technologies advance, we'll see more and more data sets where we use single-cell data, and we want to be able to normalize these values as well as our bulk RNA-seq data. Single-cell data, when compared to bulk RNA-seq data, has some more unique properties that we need to account for when we're carrying out normalization. Typically, we'll see a lot more zero counts as compared to our bulk RNA-seq, and our values tend to be in a smaller range, so our smaller differences between values can sometimes be lost if we're applying these same bulk RNA-seq methods to single-cell RNA-seq data. This means that our methods that we've discussed already are somewhat less applicable, and we need to take these into consideration. Here on the right, we're looking at a figure from a paper which is trying to illustrate exactly that concept, that where typical RNA-seq data analysis won't work as well for single-cell data. I'll draw your attention to panel 1C. Here we're looking at the relation between total cell UMI counts and the gene UMI counts. Essentially, for genes that are more expressed, we tend to see those genes more in cells with higher counts as well. So we want to normalize for that, where we're taking into account our library size differences in these single cells. This leads us to panel 1E, where essentially after some normalization, taking the log transformation with a coefficient to account for those zero counts, and then and comparing these values, we would expect to see basically an equal distribution of these different gene expression levels across our different cell groups that we divided here. But we can see the genes which are with typically higher expression values actually aren't normalized as well as these genes with smaller values, or these lower expressed genes, which can be an issue as we're trying to draw conclusions from this data. Looking specifically at some of these UMI relations, we'll see a lot of UMI counts as 0 in these single-cell RNA-seq data sets. And these values have much more discrete distributions, mainly because we're looking at very low UMI counts in most of our samples. So as we perform this transformation, we actually get these more discrete values, which are less useful to us. So we need to take into account those 0 counts across our different single cells once again. We see these more discrete distributions in the single cell data, and although our RNA-seq data actually has some of the same properties, they're less noticeable just given the difference in scale of data that we're working with. So in this case, for our very small populations, our differences in data are much more noticeable. This brings us to some of the existing normalization methods. Essentially, there is no one correct method for normalizing RNA-seq data, or single-cell RNA-seq data. This figure on the right here is from a paper trying to quantify the success rate of certain RNA-seq, single-cell RNA-seq normalization methods, where the authors devised a number of metrics measuring essentially a score of how well normalization is performed across these different data sets, and then trying out these different normalization libraries on each of these data sets. So here we can see for our first and second data sets, our far right normalization method, FQ, performs really well. It's ranked highly on this metric score that they've created. But for our third data set, this is actually scores much lower and is not the best method according to these metrics. Essentially, what we need to consider when we're doing single-cell RNA-seq normalization is that different libraries take different things into account. And depending on the type of data that we're looking at, we may want to account for those differences with different libraries. So often we can try a few different libraries and compute some measure of the effectiveness of the normalization. There are a number of libraries already available that are based on some bulk RNA-seq normalization methods, and then different models that use different types of regression or pooling to account for our 0 counts between our different single cells. Now we will take a look at some real microarray expression data. We'll take a look at sort of how the distribution changes as we apply each of our different normalization methods that we've discussed today. And we'll see how these values shift as we go through each step. So we'll be taking a look at microarray expression data from this GeoStudy, 58324, which is comparing gene expression profiles from a BG1 NIEHS cell line that was believed to be derived from this BG1 cell line, but the researchers actually found it had more similarities to this MCF7 breast cancer cell line rather than the original ovarian cancer cell line. When we're looking at our original intensity values, we're ranging from just under 1 at 0.94 to over 117,000, which if we're taking these raw values aren't significant or aren't especially useful to us. So as we're looking at these microarray expression intensities from these 18 samples that we're looking at, we want to take into consideration what each step of our normalization is doing. So here we go from our distribution on the left with our raw intensities and perform our log transformation. In this case, the coefficient wasn't added because we had no 0 values, so we wouldn't get any errors. But this leaves us with this distribution with our kind of peaks on the left and right, um, and a kind of almost bimodal distribution emerging in the center, um, with a peak of a distribution sort of crumpling in. Then when we take a look at applying our gene-wise z-scoring that we talked about earlier, our quantile normalization, our median polish methods to this log transform data, we'll see how our distributions change. And with our gene-wise z-score, we get this more normal distribution once again, where we can take a look at specific values that are more highly or lowly expressed than we expect them to be. Again, to note, for each gene, we will have a mean of 0 and a standard deviation of 1 for each row in our matrix there. After performing our quantile normalization, we get a more smoothed out version of our log data where our peaks are kind of diminished somewhat as those extreme values from are averaged on both sides. And then finally, our median polish, again, we see this distribution of residuals values that is centered on 0 and gives us some measure of, again, which residuals are actually, which gene sample relations might actually be significant. Again, we will also discuss our highly variable RNA-seq data, where we're taking a look at our libraries with with much smaller overall counts versus much larger overall counts, which again is from the study where we're comparing our peripheral blood cells and mantle cell lymphoma tumor cells. So part of what we did earlier was looking at our distributions of each sample after we do our log and quantile normalization and seeing how those distributions are more closely aligned after we perform this normalization. So in summary, we discussed why sometimes we need to normalize our data to get rid of inherent technical bias in our data and preserve our biological effects. We're going to standardize scales between samples versus looking at very small and very large values. We're going to be able to compare those more accurately. And we want to adjust the differences for library composition, size, and occasionally gene and transcript length as well. The methods we covered are z-scoring, specifically gene-wise z-scoring, quantile normalization, median polish, RNA-seq count normalization with our library size adjustments, and our DSeq2 median of ratios normalization method. These are a bunch of additional resources that can be useful to better understand these topics. They should all be linked in the copy of the slides that you got, where you can take a look at specifically each of these methods, and some of our single-cell RNA-seq normalization methods as well, in greater detail. There's also a notebook where we'll take a look at, again, our microarray expression data that we covered a few slides ago and see how these distributions change. These are all of our references. And that will cover it for our normalization lecture. I'd like to thank Dr. Mayan and Dr. Lockman for all of their help in creating this lecture. And I hope you've all learned some useful information about our data normalization.